Hi, I'm Srinand Paruthiyil. Today I'll be talking about my project, the assembling of the histoplasma encapsulating genome using optical mapping. It was a project done here at the CVRI in the Quack Lab. My mentors were Yvonne Lyon, Angel Mack, and we had collaborators from the UCSF Department of Microbiology, Srinand Paruthiyil, Mark Voorhees, and Anita Sill. So what is histoplasma encapsulating? It's a pathogenic fungus that's traditionally found in bird and bat droppings and enters the human body through inhalation. It primarily affects the lungs, where it can be ingested by macrophages and spread to the rest of the body. It's mainly dangerous to those with compromised immune systems. However, with 70% of those in the Midwest of the United States infected with this pathogen, it is of clinical importance. So we should sequence the genome so we can study the genes and obviously the transcriptional features. However, there's a problem with that. We have sequenced the genome, and there is a huge mess. This huge mess consists the entire genome. We know the specific letters, the ACTG. We don't know their order. We know that chromosome 1A exists. We don't know where it is, nor the orientation in the genome. So here's a flowchart for our project. We're going to use optical mapping as our means to address this problem. We're going to have long optical mapping contigs labeled with patterns of nicking sites, and we're going to align those optical mapping contigs to the nicking patterns in the genome, and we're going to bring together the pieces of the previously sequenced genome. So here's the creation of the optical mapping contigs. We're going to nick, label, and repair the DNA in specific sites. So here, every time GGHE-TCG appears in the DNA, there'll be a nick, and at that nick, there's going to be a fluorescent nucleotide. Next, our machine Iris will image the DNA, and it'll detect every time a fluorescent nucleotide appears, and we'll have pairwise alignment of all those DNA molecules. So here we have a specific region of the DNA, and this constitutes all the DNA molecules in that region, and we're going to have a pattern which makes a barcode-like optical mapping contig. So we then align that optical mapping contig to the previously sequenced genome. So the nicking sites for the previously sequenced genome are produced in silico or on a computer, and here we can see optical mapping molecule 16 lines up the previously sequenced genome contigs 104, 154, and 160, I think. So here are our results. We have a de novo assembly, which has 28 de novo contigs aligned to 101 out of the 172 sequencing contigs. So we have 70% covered the whole genome. So in conclusion, we have an accurate assembly of the majority of the genome, and we have found that the use of nicking sites as a genome motif is very effective in de novo assembly. So we see that large molecules can bridge together large parts of the genome. However, we have also found that fragile nicking sites hinder proper mapping of molecules. So when you have many, many nicking sites right next to one another, the DNA will fracture, and you won't get a proper analysis of the pattern of the nicking sites. And if we can find a way to combat that issue, we'll be able to have longer molecules that will span more of the genome. So in future directions, we should use a different nicking enzyme to cover contigs without any nicking sites. That last 30%, a lot of those sequencing contigs don't have nicking sites for the specific nicking enzyme which we have used. So if we use a different nicking enzyme, we should be able to label those specific sequencing contigs. We should also resequence the genome to have more accurate nicking sites in the sequencing contigs. And as always, aim for longer molecules to map across larger regions of the genome. So I'd like to thank Richard Shapiro, Julia Yee, and Max Horvath, and all of those associated with the Lowell Science Research Program for giving me this awesome opportunity to work at UCSF. Also, I'd like to thank Kuyan Kwok, Angel Mack, Yvonne Lai, Catherine Chu, and Annie Poon, and all the other members of the Quack Lab for mentoring me throughout this experience. This has really been awesome. I've learned so much about bioinformatics. It's like the coolest thing ever. So thank you so much.